So I'm David. So today I'm going to talk to you a little bit about ChromatinState and really about analysis of histone modifications. So I think, and I actually hope, it's overlap with some of the things you've heard, both from Lisa and Ross, because we're probably going to tell you some things that are similar and maybe a few things that are different. But you know, the old saying, tell them once, tell them twice, and maybe by the third time they get it. So we'll see. So yeah, there's probably going to be some overlap, but redundancy is good, at least for me when I'm learning. And so my lab actually focuses primarily, for CHIP at least, we focus primarily on histone modifications. I don't do that much transcription factor ChIP-seq anymore. I did a little bit as a postdoc when I was with Bing Ren, but mainly I did histone modifications in my own lab. Continues to do that. So yeah, so ChIP-seq, for transcription factors provides valuable information about how a transcription factor might regulate gene expression. However, that's probably pretty relevant to that transcription factor. And so I think one of the great things about histone modifications is that they provide, kind of information on a genome in terms of like genomic annotation. And so you may have heard a little bit about this, but I'll go through some more of that in more detail. And they may start to tell us some aspects about mechanism of regulation. I think some people are very reluctant to kind of link histone modifications to mechanism, but I think we might see that there's really something to that. My general experience, I taught this class last year, is that people come from different backgrounds. Not everybody is maybe not even interested in histones and histone modifications. But I like to make sure when I talk about ChIP-seq and histone modifications, everyone's on the same page. And the best way to do that is probably right here. And let's get started with just kind of the basic structure of the nucleosome, which is made up of 4 histones that occur in octamers. So you have 2 copies of each one, and then you have the H1 linker here. And then we can get to histone modifications. So the histone modifications actually take place on the tail of the histone. So the histone folds into a particular structure, and it's not called the tail because it's the C-terminus. It's actually called the tail because it's the unfolded. It's actually the N-terminus of the protein. And so this unfolded structure kind of hangs outside of the folded region of the nucleosome. It's kind of this floppy structure. And this is where most of the histone modifications occur, as you can see here for H3, H4, H2A, and H2B. And most of the histone modifications I'm going to tell you about today are actually focused on histone H3. This is where most of the information in the field resides. But there's a wide variety of histone modifications. So I have an annotation slide in a minute, but these sites can be acetylated, phosphorylated, methylated. When they're methylated, you can have a mono, di, or trimethylation. If you're at a lysine, if you're at an arginine, you can have a mono or a di methylation. And if it's di, it could be symmetrical or asymmetrical. And so there's ubiquitination, crotinylation, butylation. Some of those lead to acetylation, so it's how different they are is up for discussion. And then so all of those marks can occur on largely they happen on these lysine and arginine sites, or serine and threonine if it's phosphorylation. But most of the modifications are occurring on lysines. And so you can imagine now you have these modifications occurring on a histone tail. You have multiple histones in the nucleosome, and then you just keep adding one histone after the other. And now you start to get some complexity to the number of histone modifications. And so there's interesting questions about what does that mean in itself. So just a little bit about nomenclature, because you'll probably see this on a lot of my slides, and then if you're reading the literature in this field, it's a pretty common annotation. So histone H3, so when you see H3 in the name, this just tells you which histone we're talking about, 3, 4, 2A, 2B. Which amino acid are we talking about? So here, so we're on histone H3, we're at one of these lysine residues. And then the number that comes after the lysine, so for example, if it's 4, then that means we're talking about, so we're on histone H3, we're at lysine 4, and then of course the modification. Are we talking about acetylation or methylation? And then mono, di, tri. So when you're reading the literature, you might frequently see something like H3K4 trimethylation, and probably in the context of this talk, because I'm largely going to focus on histone H3 modifications, I may forget to say H3, which is common. So if you hear me say things like K4 trimethyl or K4 monomethyl, you should assume I'm talking about marks on histone H3. This is where kind of, again, the focus in the field, most of what we know are about impacts on annotation of transcriptional regulatory elements really focuses on histone H3. So once you become interested in histone modifications, here's kind of a general ChIP-seq flow. So you take your cells that are growing in a dish or tissue from your animal of interest, and you cross-link this. So again, Lisa may have gone through some of this with you. This is to ensure that the DNA is cross-linked to the protein. We then isolate chromatin from the cells and sonicate it into small pieces, hopefully around 200 to 400 base pairs in size, so 1 to 2 nucleosomes. And then we can ChIP for antibodies very specific to those histone modifications. So we actually test all of the antibodies in our lab to make sure that they are specific. So for example, if I go back a slide, I guess, how do you know that your antibody is truly detecting H3K4 trimethyl but not H3K4 dimethyl? It's the exact same amino acid. The surrounding amino acid sequence is the same. It's just a difference between the mono, di, or trimethylated state. So we actually test all of our antibodies that we order into the lab to make sure they're specific, to try to ensure that we're not getting cross-reactivity of our antibodies, so that when we pull out regions with ChIP, we really are going after that particular histone modification. But after that, it's pretty much the same as for transcription factors, right? You have an antibody that targets a protein of interest. In this case, it just happens to be a modified protein, a histone. And then you pull the DNA out, you isolate the DNA, you sequence it, align it to the genome. We're going to go through peak calling and enriched regions and all of these aspects of visualization, peak-peak comparisons, annotations, and sequence analysis. So again, grow the cells, cross-link them, ChIP, lyse to get everything out, sonicate, and then you can basically proceed to the general pipeline. But I will point out that there are some options to modify the protocol. So one question that some people ask are, should you cross-link or should you do what's called native ChIP? So native ChIP means you're not going to actually cross-link your cells. And so this also then determines whether you should sonicate or emanase. So emanase, monococcal nuclease, eats DNA, so it makes a nick in the DNA and it eats back until it hits the protein. So if you treat chromatin isolated from your nuclei with emanase, then those linker regions between the nucleosomes will get eaten away, right? So that DNA will get eaten away. And so if you were to run that DNA on a gel after an emanase treatment, you should see kind of, around approximately 150 to 200 base pair band, which is the wrap of DNA around a nucleosome. The wrap of DNA around a nucleosome is 148 base pairs. And so that wrap around the nucleosome is actually enough to maintain that protein-DNA interaction. And so if you're ChIPing histone modifications, you don't actually need to cross-link your cells. You can isolate chromatin, treat them with emanase, ensure that you've used enough emanase or digested for enough time that you're getting predominantly mononucleosomes. You kind of want to see this ladder of mono, di, and tri, and it's fading as you go up the ladder where it's almost hard to see the trinucleosome band. And then you can just take that material and ChIP it using roughly the same protocol. And then I think the other, so again, here's I guess what I just said. So non-cross-link, you must use emanase. If you sonicate, you're pretty much going to blow the nucleosome apart if it's not cross-linked. To a certain extent, if you cross-link to the same extent that we do to break up chromatin, or if you sonicate to the same extent as we do for cross-linked cells. And then I'll show you, so here's just a little data from some pilot studies we did at some point in the lab where, so these are just 2 tracks of H3K27 acetylation, and we'll come back to what these modifications mean, but this is ChIP-seq data on a particular cell line, and it was treated with emanase, and the track below, I think is just ENCODE data. So we get pretty similar patterns of enrichment, whether it's sonicated or emanaced. Oops. And then this was another, these are just two other histone modifications of emanase treated chromatin. And so you can get very good resolution and great chip with just using emanase if you need to or want to. And some people have argued it begs the question, do you actually need to cross-link if you're using a histone modification? And there are reasons why, especially if someone's shipping you cell pellets, halfway around the world or across the country. Cross-linking, I think, is a good idea because you really ensure that those nucleosomes stay where they should be and everything works well. But you can also emanase cross-linked chromatin. I'm not showing you that here, but in this particular browser session, there's actually a track, and they look virtually identical when we used emanase on cross-linked and uncross-linked cells. Oh, I did want to say one more thing. It's not in my slides, but so I think it was about a year, and it relates to this idea of emanase. So about a year and a half ago, there is a paper for, in case anyone's interested in this particular topic, there's a paper from Steve Henikoff's lab at Fred Hutchinson Cancer Research Center that I would direct you to. And they did an experiment. They were doing a lot of emanase, what's called emanase footprinting to know nucleosome positioning across the genome. And they decided to just do an experiment where they isolated fragments from their emanase digest that were less than 150 base pairs to see what was there. And it turns out they get transcription factor binding site footprints. It looks just like a DNase hypersensitivity map. So you could treat with emanase, isolate those very small fragments that you really can't see on a gel, and you'd have DNase hypersensitive footprints. This then led them to do an additional experiment, which was, could you then chip for those transcription factors in emanase-treated DNA to get very high resolution chip-seq data? And so they have a method, I think it's called organics or something like that. Again, I would just direct you to the Henikoff Lab website and you'll find the papers. So if you're interested in kind of high resolution chip-seq, there's a number of methods. I think, Ross, if you haven't talked about them, I think you might mention this as well, like exo-chip and these other approaches.
We were talking about chip-exo earlier.
Yeah.
About that,
yeah, so this is just one more approach that you can add to the list if you, but I believe one of the interesting things from that paper is, again, it's been a while since I've read it, but I believe they suggested that you could actually chip those transcription factors without cross-linking as well, if I remember correctly. I don't know if do you remember that?
I don't, and this is really, I'm glad you're bringing this up, right? Because cross-linkers can introduce artifacts, right? So trying to do a cross-link-free thing, and you're saying that histone modifications. Yeah,
so for histone modifications, it's pretty well established, but I believe that was one of the points in that paper was that they could do the TF chip as well without cross-linking. So for those of you working on transcription factors. Yeah. Did you have a question?
Ross, now is advantage to not cross-linking. So if we're looking for histone modifications, why would anybody cross-link? What's the downside of not cross-linking?
For histone modifications, what's the...
So you said that works pretty well established, and it's obvious that if you don't cross-link, you don't get the artifacts.
Right. So, no, I think, to me, I can't really think of a true again, for the most part, for us, like, if someone's shipping us samples and things like that, I feel more comfortable if I know from the moment they isolated the cells, those nucleosomes are held intact, right? So I just like that idea because then they're going to ship them, I'm going to thaw them and do all of these things to it. But even, I have to admit, even though it's kind of one of these things where, if it ain't broke, don't fix it, kind of. Even in my lab today, even though we've done these tests and shown that we can emanase, and we still cross-link, even on like cells in culture. I think it's just, the protocol works and we don't change it. And it's an aspect of science, right? That you kind of feel comfortable. It's like, that's what someone taught me when I did CHIP, and that's what I taught people in my lab when we first set up CHIP. And then I told someone to go try this experiment, and it looked fine, and we didn't adopt it. But you can. So just tell your boss, or if you're the PI, just tell the people in your lab. And it's faster. I mean, you can basically do an emanase digest in about 7 minutes, depending on how much emanase you use. So it's pretty robust assay. So, and yeah, and you don't have if it's going to introduce any cross-linking artifacts. Sometimes, one aspect of cross-linking that this was something even Lisa and I were talking a little bit about at lunch was, does it introduce kind of a shadow effect of not the site you're looking at, but what's looping in to interact with it? So if I'm looking at distal enhancers, there was this question when you could see weak Pol2 signal, was this due to Pol2 being at the promoter and looping in? Some people have now shown that they can see Pol2 directly at enhancers and eRNAs transcribed there, so it suggests that Pol2 actually is binding. But this was a question in the field at some point, is this weak Pol2 signal at enhancers really due to Pol2 binding an enhancer element, or is this kind of there may be a better terminology for it, but some kind of shadow effect from cross-linking, and you're just picking up whatever proteins were bound to the promoter, they weren't actually bound to the enhancer. Lisa?
Last time, we haven't done mRNAs in our lab, but we've talked about it a few times. Last time I looked into it, it was one of those really complicated things where you have to like dialyze afterwards or something like that, right? Do you still have to do that? So you say 7 minutes, but you can't just go directly.
No, well, so again, I think it depends. So the original protocols we used in my lab as well were these dialysis protocols, where then you dialyze your chromatin afterwards. But I think in the papers from like the Hinikoff lab, they actually go from emanacing to chip. He actually, in his protocol, he actually does a brief sonication to help solubilize chromatin. It's very brief. It's not really for fragmentation in the sense of breaking DNA. It's to kind of break up dense heterochromatic regions. But yeah, so in his protocols, they don't use dialysis.
Okay, so it's more straightforward.
Yeah, I think especially if you're not, dialysis can also help though if you need to change volumes too, because you can use those really small mini chambers to get to reduce your volume. All right, so I just wanted to run through, like I said, just a handful of histone modifications, some of which you may be knowledgeable of, but just kind of run through kind of why we look at them, or maybe a little bit of historical perspective on why we look at them. And as I said, I think histone modifications are great genome annotators. And so which histone modifications do you want to choose? And while this picture down here isn't annotated yet, what I will tell you is that if you don't already know, that there are histone modifications that can tell us where Pol2 bound promoters are in the genome. There are histone modifications that mark transcribed gene bodies. And then there are histone modifications that mark distal enhancer or proximal enhancers as well, but will mark enhancer elements in the genome, right? And so we'll kind of go through some of these marks. So the first one is, again, this histone H3 lysine 4 trimethylation, so H3K4 trimethylation. This marks Pol2 bound promoters, and it has been shown that it can mark the promoters of messenger RNAs, link RNAs, and unprocessed microRNAs. These promoters are not necessarily active, and so what I mean by that is originally in the field when K4 trimethylation was described, right, this kind of depends on how you do your analysis, right? If you say how many actively transcribed genes are marked by K4 trimethylation, then it's almost all of them. And so it was kind of referred to this mark of like active promoters. And then, more studies came along and said, well, anywhere between 50 and 70% of promoters might have K4 trimethylation present, and even Pol2 bound to them, but they're not actively, Pol2's not actively elongating through the gene and transcribing that gene. So that's why I point this out, that to say that it's marking active promoters, I think is not technically accurate, at least in my opinion. But how does it function as a genome annotator? So here's a paper from Bing Ren's lab as part of the mouse ENCODE project. This was a Shin et al. Paper from a few years ago, and they basically chipped H3K4 trimethylation and Pol2 and many other factors in the paper, but the point of this is just to illustrate that they were able to show by chipping tissues from mouse, fetal tissue, and then the adult tissue in mouse, so cortex, cerebellum, heart, lung, spleen, I don't remember what all they did, but in the end, they were able to identify 13,000 novel promoters in the mouse genome. Right? So these had never been annotated due to gene prediction models or RNA data. So it can be, I think, a great tool for annotating genomes or finding alternate. And they point out, I think, in the paper that indeed of these 13,000, there's the number there, of these 13,000 promoters or putative promoters, however you want to think about it, that more than 50% of them were probably alternative promoters. So they were near an annotated promoter, so either just upstream, but I think most of them were downstream in the first or second intron, suggesting that you have an alternative start site, which wouldn't be surprising given what we know about transcript variants, right? So another great genome annotator are marks like H3K27 Trimethylation. So this mark enriches transcribed regions. The K36 trimethylation tends to enrich at the 3' end of the gene, whereas H3K79 dimethylation also marks transcribed regions, and it tends to mark, I think, kind of 5' to mid-centric. And so if you chip all of these marks together, you can kind of see where transcribed genes are. And once again, here's a paper from John Wren and Eric Lander where they made use of K36 trimethylation and K4 trimethylation to try to find, if you're familiar with John's work, long non-coding RNAs in the genome. And so they were able to find regions that weren't annotated as promoters, and then they could see that based on the K36 trimethylation, that transcription was occurring in the region. And then when they went back and did RNA-seq, they could see that, again, they could confirm that these regions were being transcribed. And they were able to find thousands of previously unannotated long non-coding RNAs using this process. So again, quite informative about genome annotation. So the next mark that I'll walk you through is H3K4 monomethylation. So this marks what I call enhancer elements utilized by the cell. And even then, they're not, as the second statement says, they're not all active. And what I mean by that is, if you chip K4 monomethylation, you're not going to find every enhancer in the human genome, thank God. Otherwise, I wouldn't have a job, right? This is pretty much probably how I got a job, because in Bing Ren's lab, I helped generate the first genome-wide maps. And if we would have found them all, then what would I do next? So these, as you can imagine, just as transcription changes from one cell type to another, the use of the enhancer that drives that cellular specificity or temporal spatial specificity, if you're looking at development, is largely mediated by enhancer elements. And so in your cell type or tissue type of interest, you may be very interested in trying to find where enhancer elements are that are being utilized by that cell or tissue type that are telling you something about transcriptional regulation in your system of interest. But again, even though so they don't mark all the enhancers in the genome that you're working in, but as we'll come back to in a minute, they're not all necessarily, we don't think at least, active enhancers. And this is just that first paper from Bing's lab, the Heinzman et al. Paper, and this is kind of a figure flipped on its side, but this is looking at a heat map. Have you guys talked about heat maps yet in the course? So here what we've done is we've called peaks on K4 monomethylation and 2 different cell types. And then if you basically make a long list of all those peaks between the 2 cell types, and then you cluster the monomethylation signal, so each, in this case now, column, since I flipped it on its side, is an enhancer in the human genome. And so what you can see is that there's, if you're only comparing 2 cell types at least, there's a large fraction of an enhancer elements that are cell type specific. And again, if you're just comparing 2 somewhat drastically different cell types, that's probably to be expected because they have drastically different transcriptional profiles, right? So the enhancers that would drive that help drive that transcriptional difference should be marked differently in the human genome. Just to kind of elaborate on this point, again, here's another paper from the Mouse ENCODE project where they looked at enhancers in multiple tissue types. So again, you can see bone, regions of the brain, heart, liver, limb at E14, and then those same tissues at 2 months old, which they called adult. And again, here the row is an enhancer, and so you could see on average somewhere between or somewhere around 4,000 enhancers per tissue type were highly tissue specific, meaning that they didn't see them being utilized in another tissue type in the mouse genome. And then shown over here on the right is a paper from Brad Bernstein and others, where they looked at different blood cell types, so monocytes, lymphoblastoids, naive, memory, regulatory T cells, Th1, Th2 T cells. And while in the T cells, you can see that there's a lot of enhancers that are commonly marked, but you can see that there's kind of this spectrum of, enhancers that are enriched in the T helper cells that aren't really present in the memory or Tregs. And then you can clearly see differences between monocytes and B cells here and the T cells. So again, comparing these different cell types, you expect them to have somewhat different transcriptional profiles, and the enhancer maps are also somewhat cell type specific. So the next mark I'll introduce you to is H3K27. So in general, lysine acetylation or histone acetylation is kind of thought to mark what people call active or more open regions of the chromatin. So once you acetylate that histone, you're going to change the charge. This is going to allow the DNA to loosen its wrap around the histone. And now you can start to slide those nucleosomes apart. And this is probably going to help stabilize transcription factor binding at those regulatory regions. And so because of that, we pretty much find this at both promoters and enhancers. And here's where we kind of come to this idea of maybe not all of the enhancers marked by the H3K4 monomethylation are active. So the field has kind of defined active promoters as having the acetylation site, that you need to have acetylated those histones to kind of stabilize that chromatin structure. And so when the monomethylation lacks the histone acetylation, this has been suggested to be a poised enhancer. So I think an easy way to think about this is, of course, in the context of development, where maybe you want to market an enhancer that's going to help turn a gene on as soon as you receive a signal. So somehow you've pre-marked that region. Whether or not there's a transcription factor there or not, I think is still unclear to me, at least for most of the sites. But now you have something that's kind of predefined, and a signal comes in, and then the transcription factor can stabilize binding and recruit the histone acetyltransferase, something like P300 perhaps, and activate or turn it into an active state. So others have used K27 acetylation to solely map both promoters and enhancers, especially if you don't necessarily care about finding novel promoters, right? So once you find these intronic acetylation sites, there could be a question of, have you found an alternative promoter, or is it an enhancer? But many people have used the H3K27 acetylation, a single chip. Now you assume the distal or intronic sites are enhancers, and then you use already annotated transcription start sites from UCSC or your favorite annotation, gen code, to tell you which promoters are probably in an active state, right? And then similarly, this could be done, I haven't really introduced this mark, but the H3K4 dimethyl state, so in between the mono and the tri, has been found at both enhancers and promoters. So there's a number of papers that have been published that have used this as well to either find enhancers or find both promoters and enhancers. Yes?
So earlier you spoke about K4 trimethylation, are both to bound but not necessarily active promoters, right?
Yes.
So is that also true, just like the monomethylation, that if you do have H3K27 acetylation and K4 trimethylation promoter, that's considered active? Is that mark of action?
Yeah, so I believe, at least in our own analysis and papers I've seen, if there's acetylation, it better correlates with transcription, with elongation, that you are getting a transcribed region. But it's also genomics, so I bet you can find the counter to that.
And do you know anything about whether that correlates? So, sorry, let me rephrase that. K4 trimethylation indicates that pole is there, but not necessarily moving. So do we know anything about the mechanism of the acetylation, whether that in any way actually encourages, if you will, or makes pole start transcribing?
That's a good question. I don't know, because I mean you have to phosphorylate Paul, and you have to recruit the elongation factors when the recruitment of the elongation factors, if that coincides with acetylation, I don't know. Ross, do you know off the top of your head? I don't. Maybe I should, but I don't.
Well, these are real good questions, and I don't think our knowledge is sufficiently well resolved to answer some of them.
Yeah.
And actually, I hate to add more confusion, but these relationships also change from cell type to cell type. Not in a huge way, but here's one that just anecdotally. This distinction between K4 monomethyl and H3K27 acetylation is a big deal in ES cells, and you really can see those poised states.
Yeah.
And I was puzzled by that because I was coming at it from a very erythroid perspective. And I guarantee you, those maps, the K4 monomethyl and K27 acetylation, they're the same. And it's the same, and I think in a lot of mature differentiated cells that they are about the same.
Yeah.
So, this is kind of that interplay between acetylation and methylation, I think, may vary depending on, the commitment state of the cell.
Right, so that's a good point. When we first noticed this, we were one of the groups that tried to look at poised enhancers, or when I was in Bing's lab at the time, Bing Ren's lab at the time as a postdoc. And we were working in human embryonic stem cells, as Ross said, and it's quite prevalent there. So I would agree, we tend to see more poised enhancers, so this lack of acetylation, in stem/progenitors, and I don't know, even in the blood lineage, how much you guys have seen at some of the, if you look at HSCs but even, so when we looked in T cells, we've looked at T cells as well, we can see some poised enhancers in the naive T cells, just CD4 positive, but it's nowhere to the extent that we see in human ES cells. So, yeah, so let's see. And again, this was just, I think, this was the first paper, or I guess this might have been the second paper that described, well, I don't remember. It was either in this paper or the one before this where we mapped the acetylation along with the monomethylation. And you can kind of, well, it's actually hard to see, I think, in the projection, but whenever you see this monomethylation that's somewhat cell type specific as you move across this diagonal, the acetylation coincides. So again, with this idea that monomethylation and acetylation are often there together, and it can be there in a cell type specific manner. I will tell you that you don't have to chip K27 acetylation if you want to see an active histone state. You can chip K18 acetylation, and it's going to look pretty much identical. I know because I've done it. But I think it just kind of has stuck, right? It's kind of, it's a good antibody. It works well. It's, a lot of the early papers used it, and so people continue to use it. But if for some reason you chip like I said, histone H3 K18 acetylation, you're going to get pretty much an identical pattern and can make the same arguments. But again, and here kind of relates to Ross's point. So in that Farr et al. Paper where they looked at these blood cells and you compare the acetylation and the monomethylation, and here in these clusters, they're not actually clustering every single enhancer because you can't actually fit the heat map on a page if you do that. But you can see that the pattern and the cellular specificity is near identical. That when you get acetylation, you get monomethylation, right? So you really can see that. But again, I think if you're working in some kind of progenitor or stem cell type, to me at least, since my lab focuses a lot on different kinds of stem cells, it is interesting to map both of them to see, can you find poised enhancer states? To see if it tells you something about what's potentially happening at the next stage of differentiation or development. And again, here's just this idea of acetylation being somewhat, being very similar across. So this wasn't the first paper to look at this. Actually, one of the very first, or the first ChIP-seq for histone modification, Keiji Zhao's lab published most of these in CD4 positive T cells. And this is looking in, I think this figure was actually made from the H9 data that was generated. But you can see a lot of the acetylation sites look identical. But I would point out that sometimes, and no one has really looked into this, so if you're a computational biologist, I'm going to take a shot at this. No one has really looked at why sometimes the H2BK acetylation is different. But from time to time, place to place, it is, at least in human ES cells. And I don't know why. But I can tell you, I validated all those antibodies, and they are specific. So I don't think it's due to a poor performing antibody.
I just see a track this up there that helps answer a question that I didn't do a good job of answering earlier. You see the methyl CG over CG track?
Oh yeah, this
So
one.
that's what someone was asking, do you see the dips in methylation?
So there,
That's what I
there.
was talking about, you know, for kind of general marks for regulatory regions. And we're probably too zoomed out to see if there are potential enhancers versus promoters. But I just want to point out those dips, that they really are. If you can afford to get the data, it's really good.
Yeah, so what Ross is referring to, this track here, is actually whole genome bisulfite sequencing. So you do a bisulfite conversion on genomic DNA isolated from the cell, and you basically sequence the entire, in this case, human genome to actually, in this case, a larger depth than you normally would, because the methylation doesn't map quite as well as just straight whole genome sequencing. And it's true. I mean, if you ask people who do this on a regular basis, like Joe Ecker and Bing Ren and Ryan Lister and others, and now Gary Hahn in his own lab, they will tell you that with whole genome bisulfite sequencing data, they can pretty much tell you where enhancers and promoters are, because if they're in an active state, they tend to be demethylated. And so they would argue that just building this map, they can find the elements. So you can know when your promoters are repressed by DNA methylation, but you can find active regulatory elements just by looking for the lack of methylation at CG sites. So, but again, this is whole genome bisulfite sequencing, and this was from Joe Ecker's lab, and this is a 30x coverage of the genome. And in our hands, at least, bisulfite sequencing maps at about 75%, so you can do the math on the genome coverage that you need. And the cost.
Thanks for.
But in other organisms,
digression, but
yeah.
It was so pretty. I wanted to.
No, it's actually a good point. And then just lastly, I'll finish up with probably another common mark, which is the H3K27 Trimethylation. So in most cell types outside of human embryonic stem cells, at least I think, where this tends to form a pretty narrow peak, this forms kind of a more broad repressive peak or domain, and that spreads beyond the promoter, can spread upstream and into the gene body, and sometimes it has been shown to overlap proximal promoters, or sorry, proximal enhancers, which have also been described as poised if you have monomethylation and K27 trimethylation. They're rare, they're hard to find, they're in the numbers of 100 to a few hundred, but you can find some proximal enhancers marked in that fashion. But mainly, so it's either somewhat solely marking inactive genes, but it's not to be taken for a general mechanism for gene repression. If you looked at your RNA-seq data and you looked at all of the genes that weren't transcribed, and you went and looked to see if their promoters were marked by K27 trimethylation, that's not what you would see. It's not a general mark of repression. And so keep that in mind, because I think that's also a very biologically interesting question. How do you target those genes that have this modification and it's made by the polycomb repressive group, and then why just those genes? And then, probably between the first paper, so this is actually chip data, but this is just to illustrate that the K27 does coincide with the polycomb repressive group. So here's two members, SUSY12 and EED, and the K27. So even though this is kind of one of the original papers to do this, and the resolution isn't as great, it's still kind of here for historical perspective, and shows you that the K27 trimethylation does really coincide with polycomb. And then of course in this exact same issue of Cell, there's a paper from Brad Bernstein and I think Eric Lander that showed that the H3K27 trimethylation, when it's at a promoter, and you have H3K4 trimethylation, they called these bivalent promoters. So again, in human or mouse embryonic stem cells, this suggests promoters that are in a poised state. And they found that these are highly enriched for developmental regulators. Again, with this idea that you've somehow poised the chromatin structure at these key developmental genes, with the idea that when those embryonic stem cells receive some kind of signal that says differentiate, you're going to lose the repressive marks to make the lineage that you should make and lose that active mark to keep the other lineages or factors that might drive that repressed. All right. So again, I think here's just some illustrations of kind of some of these categories. So here are what I talked about with active enhancers. So this is the monomethylation track here in pink, and you can see in this block, so the monomethylation can overlap the acetylation. Over here are those rare poised ones that they're kind of proximal to gene promoters. It's where you can see K4 monomethylation with H3K27 trimethylation. K9 trimethylation, which I haven't really talked about, is a mark of heterochromatin, and you don't see it very frequently at promoters, but that's not, but you can find it, but it can mark very broad regions of heterochromatin in the genome. So this is just, again, a browser shot to illustrate, what these modifications might look like in the browser and how you can at least see their overlap. Which brings me to a key point. Before you call peaks, you should always look at your data. I've actually, on multiple occasions, had someone come to me and say, oh, do you mind, we don't understand our peak calls, do you mind taking a look at it? And I'm sure, can you set up a browser shot and we'll go through, or load it all in the browser and we'll look at it and you can actually see their chip just failed. But peak finders will call something, so you will get some peaks back. So before you go through that trouble, just look. I tell everybody in my lab, no matter what assay, just load it in the browser, look at it, make sure it looks like your experiment worked before you spend any additional time doing any downstream computational analysis. Because it's usually pretty clear when chip fails or when it works. So you can, because peak finders they'll find small spikes in your data and call those as peaks and output them, and you'll be asking why one data set gave you 3,000 peaks and the other one gave you 200, and it's like, well, the one that's 200 didn't work, and it's some noise in the data, and you could have learned that in a few minutes. This slide is, there's a lot of information on this slide, so you guys have the printouts, and these are just, again, some reviews on ChIP-seq, and they've spread over the course of a couple of years, some on kind of just the technology itself to a more recent review from Shirley Liu's lab from 2014 on kind of methods for, how you want to look at, not just, in this review, it's not just on ChIP-seq data. I think she covers DNA's hypersensitive data and other things that I'm not going to cover. But again, it's just to give you, for those of you who might be interested or kind of starting these kinds of projects, but might be slightly new to them, these are just kind of, some background reading that you may want to take a look at. I think a couple of them are, especially anything, most of the time things from the Nature Reviews Genetics are pretty thorough and a good place to start with kind of a general overview. All right, so once we've done ChIP, you've picked your favorite histone mark for one of these reasons. You want to find enhancers, you want to find bivalent promoters, because you're working in some progenitor cell type. So you're going to perform ChIP, you're going to map and align to the genome using standard alignment tools, Bowtie 2 or BWA, whatever your favorite aligner is, and then you're going to move on to peak calling. So this is a figure that's frequently used for transcription factors, right? So when you, after you chip a transcription factor, you may, it's random if you're doing singled-end sequencing as to whether you're going to sequence, to the left or to the right. And when you align your reads, you're going to get something that aligns to the plus or the minus strand. And many peak finders are going to look for this peak shift because if you're aligning to the minus strand, you should be slightly shifted from the center, and if you're aligning to the plus strand, you should be kind of over here, and you're going to merge these, and it's going to give you this kind of distribution, right? So this actually, you know, if you guys are using Max or something like that, it's actually one of the things that's built into one of the models, right? So when you run it, and I'll come back to this in a minute. But you don't get quite as clean as you saw from those browser shots of histone modifications. They're not nice, sharp peaks. So, you know, yes, even if you did an MNASE digest and you thought, great, I'm going to get mononucleosomes, so I'll get really clean data. Most of the time, you're going to get things that are spread across multiple nucleosomes, and so your peaks are going to be a little bit broader, and so you're not really going to get that clear middle of the peak, so to speak. And so that, so I think that's something to keep in mind when you think about calling peaks on histone modifications. So just how are you going to call peaks? Well, this is to illustrate an old saying. There's more than one way to skin a cat. It's Halloween, or it was. Right? So there's nobody's familiar with, am I the only one? Is it a Southern thing? I don't know. I don't know where it comes from. I didn't look up the history of it. But it's funny cartoons that I found. You know, you spend a lot of time. This is what you do as a PI. You basically just surf the web for cartoons. We don't really do work in our offices. At least I think that's what my lab thinks. I just do this all day.
It was Halloween yesterday.
What's that?
It was Halloween.
Yes, yeah. I thought it was appropriate. You know, so kind of gruesome, but yeah, it's Halloween. So I'm going to point it. So in the next few slides, I'm going to point out some things that we do in our lab. But again, really, there's more than one way and to do this. And as long as you follow some basic steps, you can probably find a paper in the literature that has generated believable peak calls using entirely different methods from what I'm going to show you, right? And so I'm just going to highlight a few approaches, some that I think are you know, pretty common, but there's definitely a lot of tools, and they never seem to stop coming. So hopefully when you perform ChIP-seq, you generate replicates, because it's science, and then you need to call peaks, right? So you probably read a paper, you may not realize it, or they didn't. So, you know, I think these are just some basic questions that sometimes you don't even think about, right? So you've generated your replicates, and so should you merge your replicates and then call peaks on the merged data set, right? Should you call peaks per replicate and then say, I'm only going to merge or use the overlapping peaks because those are the ones I'm most confident in? Or should you, you know, call, and again, I can't direct you to the papers that have done all of these things, but I know they do exist. Or should you kind of, you know, call peaks on your merged replicates, but use two different peak finders because you don't know that one necessarily works better than the other, especially if you're talking about histone modifications, which aren't these perfect, nice little peaks. And then use that overlap because, again, at least two different tools found that site as a site of enrichment in your data, and so you can be somewhat confident. And then if you're going to merge your replicates and then call peaks, how do you know you should merge your replicates? What's the test for that, right? And so one way to do it is just, you know, you can kind of ask if there's some correlation between your replicates, right? And again, there's different ways to do this. So, you know, one of the things that we do is first thing we do is we do a simple BAM correlation function, right? And this probably isn't really even, it may not be the best because you'll see, you know, BAM correlations actually give you pretty good. But if one data set is bad, you will see it. You'll see your R value drop from what you expect. So I point you to something called Deep Tools. This is something we use a lot in the lab. It's something once you get familiar with command line and feel comfortable with it, it's an easy tool. They have a lot of nice things like BAM correlate. BAM correlate will also generate a bigwig file for you if you want to post it into the UCSC browser. So if some of this stuff sounds foreign at the moment, if you're working with ChIP-seq data, this will become more and more familiar to you.
I think they have a Galaxy implementation.
Yeah, so
even if you're not comfortable
Yeah, so, yeah, I wasn't sure if Deep Tools was in it, but Galaxy has most of these things that I'm telling you.
I think the guys who made Deep Tools built their own Galaxy.
So I think, and what Ross is saying, I think is becoming more and more common now that as people develop new tools, they simultaneously develop a Galaxy version of it so that you can increase the, just the usage. But I think for people getting started, I think Deep Tools and another tool I'll tell you, or suite of tools that I'll tell you about, are really useful for people who are kind of new to ChIP-seq, but are somewhat comfortable or want to start learning kind of command line. It also, it has functions for like generating those heat maps that I showed you, so that can be a challenging thing to do. So it has a lot of things built into it. And so again, I'm just pointing out some things that we commonly use in our lab, but again, there's multiple ways you could do this. So for us, we typically take our replicates, do some kind of correlation. We've tried to call peaks on the individual replicates and then ask how well the peaks overlap. And this is actually an ENCODE practice. ENCODE now has specific parameters. You can call peaks on your replicates if a certain percentage or a fraction of the top, one fraction of the other fraction overlaps, it gets complicated, you should merge them. So we've tried that as well. And then you recall peaks on the merged data set. But yeah, so you can ask how many peaks overlap, so you can do this with some kind of intersection, whether that intersection is done through the table browser or some other tool, and I'll talk a little bit about bed tools in a minute. Or you can do a WIG correlation, so if you've made a WIG file, so Max, because I think you guys are going to talk about Max if you haven't already, and using Galaxy outputs a WIG file, I believe, and so you can do a WIG correlation as well. But you want to do something that basically gives you a good idea that your two replicates look similar, and at least for us, after that, we merge our replicates and then call peaks again. So some advantages to that is that you can see things, so things that are kind of weak in a single replicate. Maybe I'll show you an example here. So here are like 3 replicates we generated and some data in our lab. Here are 2 replicates I just loaded in the browser from ENCODE, and here's their merged data track. So when they merged these 2 files, they got this file, and so you can see, I think we ran max 1.4 on this. This is what we commonly run in my lab, and I'll explain why in a minute. But you can see, at least in this region here, there's some weak signal in this track, and it doesn't get called a peak, but it does get called a peak here, and then when they merge them, they get, you get a better signal. So you can improve kind of signal to noise when you merge your replicates. It also means if you have kind of a substantial amount of noise, you may lose things like this. But again, it's genomics. You got to be able to tolerate noise and realize that nothing is 100% efficient. But if your data is pretty good, then these peak finders do a decent job of reproducibility if your data looks reproducible. So you can then merge your tracks and call peaks on your merge track. And then, yeah, this is just from like a Pearson's correlation on the BAM files. And if they look pretty good, you'll get pretty high correlations. And then I think this is just kind of more of the same. But you will see that, this is how many peaks. We just took this data and quickly called peaks on it just to kind of see what the overlap would be. But then when we call it on the merge data, we get more peaks than any of the 2 individually. And again, I think it's because, some of these smaller things, I should have loaded the peak track here, sorry. Some of these smaller things that don't get called peaks, eventually, now that we have better signal, we call those as enriched sites. So again, merging the replicates. So again, there's a lot of tools a couple tools for how to do this. So some of the slides, I've made some notes or even I think attached some command line annotations just so that you have them as notes, but I'm not going to go walk you through them. So again, for us, if we just want to merge our BAM files and then move on, we use SAM tools, a very common tool for manipulating SAM files. So moving forward then with peak calling, again, you really just want to understand how the ChIP-seq experiment, you want to understand how it was performed. I think that's key, that you have some knowledge if you're about to analyze the data. And then there's lots of software out there, right? More than one way to skin a cat. I think these are probably some really common tools. I think Max is still probably the most common. It was one of the earlier tools developed by Shirley Liu's lab. Sizer and Scissor, I don't know if I'm actually pronouncing them correctly, are two other common tools. Another site that, so Homer is itself not a peak finding algorithm. It's actually a software suite of tools that, again, I think if you're kind of new to analyzing genomics data, there are a lot of tools that Chris Benner have developed, and he's put them all on the Homer website. He basically, his peak finder is based on the old original peak finder. But on this site, the reason I pointed out is, I'll come back to in a minute specific to ChIP-seq, but there's now tools on here for RNA-seq, ChIP-seq. I think there's a Hi-C module on here now. So again, if you're somewhat comfortable with starting to run some command line options and you're just getting started, these are very well developed tools that are pretty straightforward to run. So I would direct you to the Homer site if you're kind of getting started with this. All right, so Max, which is this model-based analysis of ChIP-seq, here's where you can download the tool. Okay, so I emphasize this, and people in my lab said I should not have done that, but we use Max 1.4 instead of Max 2.1 for Histone Modifications. And even though they said, oh, you shouldn't have really said not so extremely, I was like, but it's true. So Max 2.1 was developed, so it's the newer version, and specifically has a broad peak function built into it, right? With this idea that you should be able to better find these broadly distributed peaks. But, okay, so we haven't done this, n of 100, but what I've seen is that Max 1.4, at least when we specifically did a test for this, did a better job than Max 2.1 for histone modifications. But again, there's lots of ways to do it, and I would actually suggest that, you sit down, try it, and see what works best for you and your data, right? And the structure of your data. Does Max 1.4 work better than 2.1? Yes?
How do you evaluate what works best?
So that's a good question. It's somewhat arbitrary, right? The way I evaluate what works best is I literally spend a lot of time, one, looking through the browser. If it's something like K4 trimethylation, so how many TSSs, so those are annotated, did I capture, right? Or not capture? These kinds of things. And so and most of the time they're definitely overlapping. It's really about, on those broad peaks, does it call a continuous broad peak, or does it call 5, 6, 7 narrow peaks, which I'm then just going to concatenate together anyway. And that's really what we're talking about here, and we'll come to this in a minute, is that I don't see, or at least in general, I don't see a lot of, Max 2 is completely missing something. It's more of, when I see these broad regions, I want them called as broad. And that's maybe a bias on my behalf. Maybe there are subtleties in the data, and I should leave them as such as individual, say, enhancers for the monomethylation peaks. So that's more of what I'm getting at, is when you have these broad kind of continuous regions you can see in the browser, and it doesn't call them as a broad continuous region. But we'll come back to that a little bit more. Here's the links to Homer. And so one of the things that Homer does is that it does have several options for peak calling in the broad setting. So trying to find, like, you can set parameters for kind of general distributions of how narrow or broad you think your peak should be based on looking at your data, things like that. And so in that sense, if you're kind of new to peak calling and you just want to play around with parameters and see how it changes, you can do this in Max as well. But Homer also allows you to do this, and because I think it has a few more parameters you can set, and so you may be able to best fit it to your data. And that's also true of some of the other tools that I mentioned prior to this. Is there something? Okay, and again, that just comes to, so for Matt, it takes the map files, it creates a file for displaying these kind of, dense amount of data. These are your wigs or your bed graphs, or at some point you might make a big wig. And then again, there's some command lines down here if you're running it as opposed to if you're not in Galaxy. And then it can run with and without control. So the control, which I guess I may have skipped over early on, is this input track, what we call input DNA. So after you sonicate your chromatin, you take an aliquot of that out and you just isolate DNA from it and sequence it. There's no chip enrichment. And this kind of serves as your background, right? So you want to find enrichment relative to that background, right? So if you don't sonicate very well, you get these spikes in your input track, and then you'll also see that spike in your ChIP-seq, and it's an artifact of sonication, right? And so if you run Max or these other tools with the control, then it takes that enrichment in your ChIP data relative to kind of the background level in your control. And so, but there's, so that's what in Max, when there's no input, that's what this no lambda model is. So it's not going to, basically it's going to skip over this initial processing that it does for kind of local background estimation because you don't have it, right? And then for the histone marks, it typically runs in this you want to run in this no model. And again, that has to do with that cartoon illustration that I showed you at the very beginning when you see the positive and negative alignments, and it expects your data to look like this, with the center of that to be probably where your transcription factor is, but you're not getting that nice narrow peak, so we typically run it in this no model so that you're not trying to model for that shape of the data, right? And again, Homer does something, it does something similar. It creates these nice tag directories for you, so now you know where all your tags are in the genome, and it has a bunch of downstream applications that you can use. You can plot other signals at the same site. It will take those tag directories to say, when do you want to find nearest neighboring gene? Yes, Lisa?
Just for your information, after you told us about this last year, Chris has been using Homer, and we have it now on Galaxy, so these guys are going to be able to use it.
Okay, great. Yeah, I mean,
Not the full suite, but it's...
yeah, so I mean, it allows you to play around with some things that I think, and it kind of helps you understand how changing parameters work. And I'll show you some browser shots of that in a minute. And again, just some command lines if you want to give it a shot. Yeah, so Homer kind of does the same thing. I'll kind of skip through it, especially if you guys are going to have an opportunity to play around with it. And again, all of these peak finding tools, no matter which one you pick, there's certain parameters, again, like I just pointed out, like the no lambda, the no model, or other things. If you're in Homer, you can kind of set the distribution, what you think the width should be of peaks, to try to play around with how you're going to find those narrow versus broad peaks. And you can try to estimate that sometimes based on whether you've done these things as well. And then, so yeah, I just wanted to show you some examples. So this is, some K4 monomethylation data. And then these are several different, so you can set different size distributions in Homer. And you can see then it tries to find broad peaks in some size distributions, whereas if you're trying to force it to find narrower peaks, it might split, and maybe this should be split into multiple peaks and not be considered a large peak. So again, this is kind of what I mean by like, look at your data, how do you know if it's working, right, based on your peak finding? Just run it under a couple of different conditions and ask, do I see it? Or you can, if you get slightly more sophisticated, you can then plot signal intensity under all of your peaks and see how the signal intensity shifts. Maybe that gives you an idea of how good your signal intensity is, depending on which parameter you use to call peaks. So just to illustrate, again, these are just to illustrate some points. Here, the ChIP-seq data is not loaded in, and you can't really read this, but on here are basically Max run under, I think Max and then Max 2 run under two different conditions and Sizer. And you can see kind of as you just look at these blue ticks, we basically ran these on, on the same data but under different parameters just to get a feel for what happens. And you can see if you're too I mean, again, the data's not shown, but you can see that under some conditions you can start to call a lot of peaks, and under other conditions you may not call many peaks at all, right? So again, I would say, look at your data. Again, here are some tools that were, so Homer and Sizer and this other Bayesian change point, they have tried to emphasize broader peak formation. So this is some somewhat noisy H2 ubiquitin sites, and then this is K27 Trimethylation. And so these tools are trying to call peaks on those regions. So do you think this cluster should be one broad peak, or do you think these individual spikes are peaks, right? And that also depends somewhat on scale that you're looking at. Yeah, just take a look and see what you think if the peak finding seems to fit the structure of your data. Yes.
Sort of to emphasize what you're saying about literature data, it's sort of the things on this course. But one of the things is sort of asking, how do you tell whether you're using the right parameters or whatever for peak calling? And if you look at the data and you see peaks that you can obviously see with your eyes,
Yeah.
that is not being called by your peak finder, you probably need to tweak the parameters.
Yeah. Or if you're, if you see something that's, being called that's like some tiny little thing out by itself just because it's somewhat isolated, but it's, barely above background, but because it's isolated, so background around it, maybe you're calling too many peaks because you're calling just subtle enrichment above background. So just these kinds of things. And it's somewhat trial and error, unfortunately. There's no perfect approach.
You just look and see, the real peaks are huge.
Yeah.
So this little teeny thing down in the noise is probably not something
Yeah.
I should pay attention to.
These peaks are all, these graphs are all taking into account the signal you get from your control experiment.
So actually most of these I've just loaded in raw data for you. So usually what I ask people in my lab to do is load the raw data, right, the chip data, load the input data so you can see them separately, and then you can make an input normalized track once you see that your sonication, your input looks fine, and then you can look at input normalized data after that. So everything pretty much comes off the sequencer, goes into the browser as raw data just to get a feel for how well things are working.
And then you look and see whether you see similar patterns in your control, in which case you...
Yeah, so your control should be just kind of a flat line. You shouldn't really see much in it, hopefully. And then hopefully your replicates look similar. All right, so again, maybe in some instances you think this whole region should be a broad peak. You don't really care about the individual spikes because you care about overall data structure. So even when your peak finder doesn't call it as a broad region, you can still mark those regions as such. And some tools will do this for you, like Sizer, S-I-C-E-R. You can basically give it an option that if two peaks are within some distance of each other, it merges them into a single bigger peak, right? So maybe it called a peak, there's a 200 base pair gap, and then you called another peak. Well, maybe that 200 base pair gap isn't meaningful, and so it's really just a broader region. The signal dropped just low enough that the peak finder sees a break in signal and then calls the next region over. So you can basically then stitch together or concatenate your peak calls based on some size distribution. So we do this, we use this merging option in bed tools, and basically, and I think again, there's lots of ways to do this. This is just to kind of illustrate how we do it. But if there's peaks that overlap slightly, in this case, I guess this would be an example if you have multiple samples and you called them and you wanted to merge them. But more of what I'm talking about is an example like this, where if there's two things and there's some distance d, you can set the distance in the command line, some distance between peaks. This example, they give 10 base pairs. We tend to use between 150 and 200 base pairs. You can look at size distribution of your peaks. You can look at average distance between peaks and call some cutoff that you want to merge together. But then we merge those into a single peak call. So if once we run the peak finder, if we see peaks that are within very close proximity to each other, we merge them into a single peak. So even though the broad peak finders aren't perfect, you can get slightly broader peak calls using these kinds of functions. All right, so I added this on really just to give you an idea that these tools are out there. I'm not really going to go through them in great detail, but you know, let's say you're not just interested in enhancers. Why, I don't know. But maybe hey, I built ChIP-seq maps for 30 histone modifications. So let's say you did the same thing. You took your favorite cell type, you chipped 30 histone marks like I showed you in that big heap or that big map I had before of all those histone modifications, and you thought, wow, I'm going to have to call peaks 30 times and then find when this peak overlaps this peak for this histone modification versus that modification. Sounds like a lot of work. So if you're working with multiple histone marks simultaneously, there are tools that try to help you assess regions of enrichment without actually calling peaks. Well, this one somewhat needs a peak. And so 3 that came to my mind were ChromaSig, ChromeHMM, and Segway. These work very similarly. In fact, there's a paper from these 2 ENCODE PIs that compares them and shows they work similarly. So out of these, ChromeHMM is the most commonly utilized, and I'll show you some data from one of the publications related to this. For ChromaSig, it's more just, I think a simple way to think of ChromaSig is that it's kind of like if you wanted to do k-means clustering, right? You wanted a heat map of regions of the genome where multiple histone marks overlap, but you don't know which k-means to use or which histone mark should you start with first. So the way ChromaSig works is you could say, I'm going to take all TSSs. I'm going to drop all my histone marks into it. It's going to determine the optimal number of kind of k-means, and then it's going to cluster those histone marks grouped based on a similar pattern and break them down into however many clusters there should be. ChromeHMM is a hidden Markov model. You guys have talked about that. Segway is a dynamic Bayesian network method, which I think are what hidden Markov models are based on, but I'm sure that someone else can speak to that better than I can. And so I think, again, when you think about histone modifications, this idea of you have a signal, you have a change in state, that signal continues, and you have a change in state again. I think that's a simple way of thinking about how a hidden Markov model might work, right? And so you're going to identify regions where there's a change in signal or a change in signal state. And then after you do that, you're going to look for patterns that occur over and over and over again in the genome, right? And so it's going to do all of this for you without calling peaks per se, right? And so the idea is that you're going to take a whole bunch of histone marks, and they have, you know, signal, and some of them have different kinds of signal. Some are kind of punctate or more isolated. Some are more extended. Some are more distal. You don't find them within the gene, etc. It's going to reduce this down to kind of some simple data structure of where it finds regions that look to be enriched based on these signal state changes. And then it's going to further reduce that then into kind of this heat map scenario where now you can see each histone mark across the top. And then based on clustering of histone marks that are co-occurring in the genome, it essentially divides them into you know, promoters, transcribed regions, active distal regions, kind of repressed regions, and so forth. But even within, say, for example, active you know, intergenic regions, you're going to get, you know, monomethylation, K27 acetylation, and so it's going to categorize these as strong enhancers versus if you have monomethylation but no acetylation, it's going to categorize them as weak enhancers, right? So it's going to kind of do the peak finding for you on all the marks at once, group regions of the genome that have co-occurring patterns, and put those together into these kind of categories for you. So if you're working with multiple histone marks and don't want to have to call peaks on every single mark, then using something like ChromeHMM or Segway could be a good approach to kind of defining regions of the genome that are marked in a similar way, and then there's enough knowledge there to know if that one of those groupings has monomethylation in it. You're talking about enhancers. If it has K27 trimethylation or K4 trimethylation, you're talking about promoters, and if it has K9 methylation, you're talking about repressed heterochromatic regions, right? So I'm not going to go into these in great detail, but I just wanted you to be aware that there are kind of some additional tools out there for grouping chromatin data together. So this summary is just really about histone modifications, or peak finding, not the last part. Again, I think they provide valuable information about the localization of regulatory elements, kind of telling us where enhancers and promoters are and if they're in an active or poised state. They can start to tell us something about gene regulation. So when do we acquire these repressive modifications or lose the active modifications? Look at your data before calling peaks. Look at your data after calling peaks. And just bear in mind that even when you think you have these optimal peak finding conditions, most likely you do probably want to run one of these merge functions, or if you're using something like Sizer, which you can set that already, I think, in its pipeline, that things close by are probably part of the same overall chromatin structure. So you can kind of stitch or concatenate, whatever you want to think about those kind of regions together. All right? So I think I went a little bit long, but that's all I have. Thank you. Yes?
So a lot of times some of these histone modifications, when I'm thinking like H3K27 we see these duplets, right?
Yeah.
And so there's some reason to believe that the region between the duplets is kind of where you want to look for motifs, right?
So that is true. So what Lisa's suggesting is that you kind of see these within kind of a slightly broader region. You see these really strong peaks, and the question is, in between that little doublet or whatever you want to call it, is that kind of where your transcription factor is most likely to be residing? And you have a really strong acetylation site next to it, right? Because that transcription factor has recruited the histone acetyltransferase, and then that signal kind of fades as you move out from it. So that is true, that you want, finding regions like that may help you narrow your motif finding. So it's not something I've talked about here, but often after you've mapped enhancers or even promoters, you'd like to know or predict what transcription factors are binding to those elements. And well, if you call an enhancer that's 1.5 or 2 kb wide, you're going to come up with a lot of motifs, and they're going to be significantly enriched. So if you can narrow that space by kind of looking at your signals. So one of the things you can do, and so actually Homer is a nice function for this. You can do it in other tools, and we do this as well. But again, for getting started, Homer has a nice function where it will kind of take the center of your peak, regardless of its width, and try to, or you can have it try to find a center. But basically, if you call the center, and it will try to plot your signal intensity. And so you can see if you get that kind of little doublet appearing, and then you can focus in on where that is.
Because that should be the region that's been cleared of nucleosomes, right?
Right. So you're trying to get at a nucleosome-free region without having something like DNase hypersensitivity assay or DNA-seq or attack-seq data. There are some other tools. One that I think there's a function in Homer, but there's also a tool from Shirley Liu's lab that will try to find nucleosome-free regions, right? So with this idea that if you've emanased or done a really good job of sonicating, most of your signals should come from the nucleosome. And so when we plot these things in the browser, we kind of smooth over the signal. But if you actually plot them on a tag-by-tag basis, you really can start to see nucleosome structure. And so what some of these other more advanced tools do is they try to find gaps between that signal and your ChIP-seq data and predict regions of nucleosome, or predict nucleosome-free regions. So we've done that as well, where we've tried to predict nucleosome-free regions from ChIP-seq data without having hypersensitive data or attack-seq data, something like that, to narrow our motif analysis. But I think the other best way is what Lisa's suggesting.
So there's a workshop in there for you to just continue with what you did this morning, plus an exercise for what do you do when you got your piece. Also, the data set that we gave you for doing max can also be used for Homer in the same galaxy, so you can run them out and compare to see what piece you get. I love it. I think the best thing way for you to learn how to use these tools is to mess around with the parameters and see what happens, and then display it in the browser and take a look.
If you just load those, the peaks usually come out it's bed files, right? I think the same even in Galaxy. You just load them all in, and you'll see those bars just stacked on top of each other, and you'll see if they're broad or narrow, or if one condition didn't call a region, or something like that.